Our next presentation will be Seth D'Imperio, who is Senior Microbiologist at Environment over in Bozeman, and he's going to talk about microbial controls on Minewater pH. Seth, it's all yours.
Thanks. I hope everybody enjoyed the break and came back good and caffeinated, because I'm going to be coming at you with a little bit of microbiology and Latin names for organisms. So keep that coffee coming. Before I get much further into this, I do want to say thanks to the organizers, and thanks to the organizers over the years for this. I started with Environment 5 or 6 years ago, and very shortly after I started with Environment, I came to my first Fairmont conference, and I knew basically nothing about mines or tailings or any. There were words getting thrown around that made no sense to me at all, and this whole group has been incredibly welcoming and willing to teach, and it's just been a wonderful experience, and I'm happy to come back and talk at it here again, and hopefully spark a little bit of interest in some of the things that we're doing. So today I'm going to give you a little story about some work that we've done in conjunction with the Siboney-Stillwater folks. We were doing our work at the Stillwater site. Here, I'll try not to move too much so that I can get followed around. Out by Nye, and also importantly here at the, if I can find it, the Hertzler Ranch out here, where they have a land application disposal pond that was doing some odd things. So we went out there to help them diagnose a little bit of what was going on in their overall wastewater treatment system, but also at that LAD pond. So we got a really great introduction, or more than an introduction, from Randy yesterday on the Stillwater site and their water treatment objectives. So I won't get into too much detail there. Just reiterate that their big issue that they need to deal with is some nitrate, starting out, heading into their water treatment plants at just under 20 milligram per liter nitrate, with a little bit of ammonia in there, and a circumneutral pH. Nothing terrible, but it needs to be treated, of course, before it is released back out, either into land application or into any local water bodies. So it goes into two MBBR systems, so moving bed bioreactors as their biological treatment plants. They colloquially refer to them as the bug plants, and I just like that, so I stick with it for a lot of things. In fact, I at one point considered calling this talk a bug's tale, but then I was worried about copyright infringement, so it's told off on that. The MBBRs have plastic media in them. They look like little pasta noodles, nothing unusual there. And they feed those plants with methanol as a carbon source and phosphoric acid for the nutrients that the organisms that are in there need to perform the job that we want them to do, which is to denitrify, get rid of that nitrate that's in there. And sure enough, with the oversight of the group that's there and is taking care of those bug plants, they're doing quite a good job of that. Nitrate's way down below 1 milligram per liter. The ammonia has dropped some, and the pH, again, stays pretty neutral. So, try to point again here. Here's the bug plants. It's represented here by a single unit, but there are two of them there, and there are two cell units that are there. And Randy got into a little bit of that yesterday. And that water then flows out of there into collection pond 3. And then on this map, it just says subtly that it heads off to the Hertzler Ranch. And on the right, we see the Hertzler Ranch, or at least the parts that I cared about while I was there. It's a decent sized ranch. Here's our land application pond. So that's a holding pond prior to going out to land application, and just to the side of it here, so it's just adjacent to this tailing storage facility here. That's a lined facility, and there's a little bit of influent from that water making its way into the land application pond as well. So again, the inputs to that LAD pond are discharge from the two bug plants. Those actually get combined into a single discharge, and a tailings under drain that's a part of that tailing storage facility liner system. And the eventual application of that is either land application on some pivots that are out there, and they water the grass, and that feed some cattle out there, or percolation in some unlined ponds. But the vast majority of that is set up to go out for land application. And in terms of a breakdown of where the water is coming from that makes it into that LAD pond, the vast majority of it is coming from the bug plants. So bug plant water comes out, it's nice and clean, the nitrates come way down and gets combined, heads up to the ranch and goes into that pond. There's a small amount of septic flow that makes it into the LAD, but it's just 1% of the total water getting in there. And then that tailings under drain from the adjacent tailing storage facility is about 4% of the total water there. And all of that was working quite well up until around July of 2019. And at that point, the operators there noted what at the time was a slow increase and then a faster increase in the pH of the water in that LAD pond. And then getting out to about September of 2019, the pH of the water in the LAD pond had risen above their discharge limit, which means now they've got a pond that's filling and they can't get that water out to the pivots. They can't do the land application that it's designed for, and they're starting to run out of room in that pond. So as a temporary fix, they started treating some of that water with with sulfuric acid, which of course brought the pH down in that water for some application, but that's a bit of a Band-Aid, and no one was particularly happy about that. And that's when we got the call from them to kind of dig in a little bit to find out if there are any biological reasons that their pH is going up in a storage pond, essentially. So the first things that I look at as a microbiologist when I'm looking at an increase in pH are denitrification and sulfate reduction. So we know from the water chemistry coming out of the bug plants that, boy, there's not a whole lot of nitrate coming in there, but we did have a look at that tailings under drain to see if there's any more coming from the tailings under drain, and no, it's not really. That's a nice low nitrate concentration. There's a bit more sulfate coming out of both of those, and sulfate reduction can lead to an increase in pH, but the stoichiometrics of the amount of sulfate heading into that relatively large pond really the math didn't work out for the pH coming up into the above 9.5. We of course looked at the actual pH of that water coming in. I think maybe that the tailings under drain all by itself was raising that pH, and yeah, the pH is a little bit elevated in the tailings under drain water, but certainly not enough, especially with all of the dilution going into that. That's only 4% of the total volume going in from the tailings under drain. Not enough to push the pH well up above 9. There's some carbon making its way into the system. That's as organic carbon right there. So certainly enough to support some bacterial growth. But again, the math didn't work out on the pH going up just through denitrification and sulfate reduction. Also, there was no real evidence of sulfide in the system. So sulfate reduction kind of goes out the window there. So we decided to take a deeper dive into what organisms might be in there. And just looking at the pond itself can give us some idea of what's happening, but it's also nice to trace back and understand where those organisms are coming from, and if there is a point source for organisms that might be causing some pH increase or any changes in the system. It's certainly easier to treat at a point source than it is to treat in a large pond. So we sampled in a few places here. I've simplified things for my own good, so I know what I'm looking at here. So we looked at the bug plant influent. We looked at media in that MBBR in the upper bug plant. Effluents from both of the bug plants, the upper and lower. And then way out at the ranch, we had a look at, that's down here, this is the outflow from the bug plant into the LAD pond. We looked at microbes at the pipe from the tailings under drain. And then up here in the picture in the top right, you can see a little bit of a slimy substance that forms as that water's falling down onto the liner into the LAD pond. I know it doesn't excite everybody, but for me as a microbiologist, when I see slimy things, I get all excited. So I couldn't help myself, and I went ahead and had a look at some of those slimes. Surface-associated microbes are often quite active, and it's almost like you're getting your microbes concentrated in a slime like that. So I went ahead and took some scrapings from that as well. And then the last place that we sample here is this recirculation pump. You can see our access is actually just a little bit off camera here from the LAD pond, and that gives us kind of a bulk idea of what organisms are in the LAD bulk water there. And so what do we do once we have these samples? First of all, we had to devise a way to sample water immediately as it was coming out of the pipe. We wanted that water because it's a pretty long distance for microbes to go to make it from the water treatment plant on site all the way out to this LAD pond. And there can be some changes that go on there. So we wanted to be able to make those comparisons if at all possible. So we take those samples. For our aqueous samples, we filter them, not so much to clean the water, but to collect all of the things that might be living in that water. So we filter onto 0.22 micron filters. Then we can remove the filter material itself and extract DNA directly from those. So you see, I think that's Brandy right there, working in our BSC. That's a biosafety cabinet. We do a fair amount of our work, especially with environmental samples in this biosafety cabinet for two reasons. One, there is the off possibility that there are some human pathogens in anything that we grab, so just for safety of our operators. But the other big issue that we have is we and all of our environments are covered in all kinds of microbes, and it doesn't do any of us any good to do a lengthy and detailed characterization of a microbial community if all we're finding out is what was on our hands and gloves at the time that we sampled. So, these things go into a nice, clean, a clean environment. We take whatever samples we have, if they're scrapings or filters from the water. We have some DNA extraction methods that we have. They're mostly modifications of commercially available DNA extraction methods. Most of the methods that are out there were developed for things like rat tails and blood samples and that sort of thing that are very high biomass. And then we come to a system like this outflow or the pond water itself, and there just aren't that many microbes in there. So we need to go a couple of extra steps to both concentrate the biomass that we're working with, and then to make sure that we're getting as much DNA out of there as we can. Even with that, we still don't have enough DNA to sequence directly, and so I've lost myself here. We use PCR to amplify a very small fragment of the total DNA that's in there. So by amplify, I mean we take the 2,000 to 20,000 copies of that DNA that are total in there, and turn that into a couple billion copies of those DNA fragments. Once we've got that, we can move on to sequencing. We do our sequencing out of house. We have a few vendors in North America that we work with. That's worked very well in our lab up until the fun of COVID. At that point in time, the laboratories themselves oftentimes had some issues with staffing and keeping their people healthy and in there. They were also under an awful lot of demand, especially early on with COVID, to keep up with the volume of samples that were suddenly coming in to do viral sequencing. But we do manage to get that done. Sequence comes back to us raw and ugly as a whole bunch of colored peaks. We clean that up to make sure that we're looking at our own environmental samples, do our own QA and QC in-house, and then take that out to community characterization here. So we've gone from a sample with just a few microbes in it out to being able to find out who's home in all of these different samples. And so to answer that question, who is home? Well, it depends on where you are. The influence to the bug plants had a fair amount of diversity here. So we're looking at about just under 200 genus level organisms in those samples there. And then we get into the bug plants themselves, where we're feeding them with methanol and phosphoric acid. And so we've got nice high diversity in those systems. As we make our way further downstream toward the LAD influence, there's the main influence. We do lose a little bit of that diversity. That's to be expected. The tailings was a little bit different. It's relatively low diversity. There's not a whole lot of nutrient to work with in the tailings underdrain, and so the diversity of the system reflects that. The scrapings, for as excited as I was about them, really didn't represent a whole lot of microbes in the system. And the LED pond itself was not particularly diverse, but there's a big change in the makeup of that microbial community as we get into, from the water treatment system and then into the LED pond itself. And that has to do with this big switch away from bacteria and toward eukaryotes. And what kind of eukaryotes? So the eukaryotes are the higher life forms, things with a nucleus. We're eukaryotes. We got, plants and things are eukaryotes, and that includes things like algae. And what we had in that signature were a whole lot of chloroplasts there, which led us down a slightly different path. So rather than just sequencing genes that are involved in bacterial metabolism, we added in a gene called the ITS gene that is helps us to identify eukaryotes like algae. Just so that I'm not bad-mouthing all of the bacteria out there, we're outnumbered, so it's best not to get on their bad side. We do, in general, like to see all of this diversity. If you're looking at these figures and all of these pretty colored bars, each one of those colors represents a unique genus level taxa, is what we call them. In a bug plant, in a biological treatment system, diversity is our friend. And that group of organisms that's in there doing denitrification and cleaning that water and getting it ready for land application or whatever the discharge is, they need to be both active and resilient. Anybody who's spent any time in a water treatment plant knows that they are never consistent and they're never turnkey, and they always need some changes, and there are temperature swings and influence swings and chemistry that changes the whole time. So the more organisms we have in those systems that can fill that niche of doing denitrification, the better. So, the upstream stuff, to me, it looks great. We're happy to see lots of diversity. It means that those systems can put up with upsets and changes in influent really quite well. And then we get out to the LED pond, and that diversity's dropped way off, and we've suddenly got this whole new group of organisms that are eukaryotes, and they're very likely algae in that system. So whose home, you were asking? I know you weren't. Up here at the bug plant influent, we have a whole bunch of organisms called Comomonas. In fact, that's better than 50% of it. So again, we start out with not a whole lot of diversity. We get more diverse as we move into the bug plants. That media covered in Methylotenera, Rhodobacter, and Comomonas still. They stick around for most of this process. As we get further downstream, we see more Methylotenera, and we'll get to what these things do in just a moment. Same crew here at the entrance to the LED pond, which is encouraging to me. It means that that's a nice, robust community, and that they are still making their way through the whole system. The tailings underdrain is different. You see Desulfobaca here, and of course Comomonas. I should note that Comomonas is just about everywhere, including all over this room. Don't worry about it. But then we get to the LAD pond, where we pulled these samples from the recirculation line, and we've got this group called Mediophyceae, along with some Flavobacterium and Comomonas. And that's a big change, and it's not just that that organism is there, but boy, that's an awful lot of the community are those Mediophyceae. So what are these doing? Well, frankly, they're doing what we want them to do for the most part. The Comomonas here, they're involved in nitrification and denitrification. That's great. We've got ammonia and we've got nitrate that needs to be dealt with. Once we move into the bug plants and even further out the line, it's dominated by denitrifying organisms here. Methylotenera, unsurprisingly with the name, they can take methanol, which they've been adding to the system, and couple that to nitrate reduction, and that's the organism that's doing the heavy lifting in the bug plants, and it's great to see it there in such high numbers. There are some sulfate-reducing bacteria that come in from the underdrain at the tailings facility that's next door. Not particularly surprising given the amount of sulfate that's in that water. And then we get down here. Then yeah, Mediophyceae are algae, and they're phototrophs. They're in fact, they're very pretty green diatoms. And in a pond system like that, where there's a pretty good surface area and a fair amount of light heading into it and reasonable temperatures most of the year, it's not surprising that there are algae in that pond. But what does that mean for pH? Under normal circumstances like what we're breathing, we've got CO2 and we add some water to that, and the trees and our grass and everybody else out there is perfectly happy to turn that into some sugars, and we get oxygen out of it and everybody wins. In a dissolved system though, we've got bicarbonate. And when we put that into photosystem 1, photosystem 2, at the end of it all, sure, we get some sugars and we get some oxygen, that's great, but we also get some alkalinity out of it. And with enough growth and enough of the nutrients that they require, we can go from relatively neutral pH in an unbuffered system or a lightly buffered system up into pH around 9.5 all the way up to 10 if they're left unchecked. This is not my imagery. I did not get these under this scope, but this is the type of organism that we're looking at. They're actually really pretty to look at under a scope. So what do we do about algal growth if we've got it in a big pond like this? Well, the things that control algal growth are light and temperature, enough CO2 for them to grow, the right nutrients for them to grow, and in an extreme case, if we need to, apply some algicides to them. Light's a tough one to take care of in a big pond that's out on a ranch. Temperature, same deal. It's a pretty big volume to start messing around with the temperature for, and most algae have a wide range of temperatures that they can grow at. There's going to be some seasonal variation, but the ones that we're dealing with here are actually quite happy when it's cold. CO2, boy, that's diffusing right in. That's another tough one to control. The nutrients, on the other hand, that's something that can be controlled because at this pond, the bulk of the water is coming from a water treatment facility. So they do know what nutrients are going in and which ones are coming out. And algicides can be used, but they have some temporary efficacy. They need to be reapplied often. And oftentimes they're regulated in discharge water. So it's not the most ideal situation. So let's focus in then on the nutrients that were going into that pond and how they were accumulating. The blue trace that we see here is, oh, thanks, you were on it for me. That's the pH that we've already looked at. If we look at the accumulation of phosphorus in the yellow, that was coming up also. There's a correlation between the amount of phosphorus that's in the pond, at least for the early stages of this, and the pH rise. So what we did here, had a look at that, saw that rise in pH, saw that coinciding with an increase in the amount of phosphorus that we were detecting in the water in the pond, and suggested at that point that they minimize the phosphorus addition at the bug plants. It's very easy to have an operating bug plant with a nice, robust microbial community doing everything that you want and say, well, when we add phosphorus to this, it works great. Let's add more phosphorus. We really need to link that phosphorus addition to the amount of nitrate that needs to be treated in there. And that way just optimize the entire time. That way there's not as much phosphorus making its way downstream and into the pond. And then of course, the other thing that needs to happen is that we need to continue monitoring here. And that's something that the group at Stillwater, of course, was doing. It's part of their discharge and they also want to know how well things are working. So we suggested that they continue that monitoring, but also continue to monitor the microbial community out there. And after some time with the phosphorus low, and there is a little bit of lag time there, and I'll address that, the phosphorus doesn't leave the system all that readily. It gets taken up by the organisms that are in there. Those happily grow, and when they die off, they just release that phosphorus back again into the water. It takes quite a while for either the biomass to sink down to the sludge that's in the bottom, or to have been taken up by macroinvertebrates that are in there and actually removed from the pond. So there's sort of a reservoir, but eventually, if you decrease the amount of phosphorus going into the pond, it becomes limiting for the algae that are in there. The algae stop doing their thing, the phototrophy slows down, and the pH goes down. And we can see that in some quantitative PCR that we did here. We were actually looking at the number of phototrophs total. So this is now looking directly at the number of chloroplasts that are in samples that we take there, and we see that drop down, and it coincides with the drop in pH that happened in June and July, and you can even see it starting to come up just a little bit in July of 2020. The The big take home here, though, is, just some optimization, not going overboard, and keeping an eye on nutrients can keep a pond like this healthy. It allows them to continue to use it for land discharge without having to treat with sulfuric acid. No need for algicides in the system, so long as they are keeping an eye on their microbial population and just keeping some general tabs on it. And with the good group that's out there that is keeping good close tabs on what's happening, that's exactly what they've got going on right now. With that, I'd like to thank our group at Environment, Lisa and Kyle Miller, who helped me out with a lot of sampling there, and Lexi Gilbertson, who did a lot of DNA extractions for me, instrumental in doing that. And of course, the group at Sibanye-Stillwater, Randy and Amy and Eli and Billy, who got us out there on site, walked me through their system countless times so that I understood exactly where I was sampling from, knowing full well that I was going to turn it into ugly block, the flow diagrams for myself, and have been keeping us up to date on what's been going on in that system since. And with that, if anybody's got questions, happy to answer.
Questions for Seth?
So I'm just curious on the decisions related to the different inputs and particular light. It seems like maybe there are physical measures you could use to control light, and I guess in the end it's probably a cost balance and dealing with the nutrients and stuff. So did you walk through that type of analysis with it?
Sure. Yeah, the question is whether or not we considered physical methods to control light heading into the pond. It is definitely something that we spoke about with the group there, and I think it's still something that it's on the table if they have this issue again. There are, any number of different ways to do that, anything from covers to the floating balls to the, and all of those have some merit, they needed to take care of things relatively quickly and not have it be a large undertaking at the time, and it was also kind of the middle of winter when we were first dealing with this. So, controlling phosphorus was something that they could do readily. But yeah, cover control for light incidents there is certainly an option. Yep.
My question, well, first of all, we do a lot of sampling in rivers in Montana, and it's nothing to see a pH of 9 or 9.5 in the river. And I guess I have to question the discharge standard. The discharge standard, we have a lot of regulators here, but I don't understand why it's that low. And also, Georgetown Lake, Warm Springs Ponds, their pHs can get up above 9 easily in the summer. So I just don't get that. And
I'm with you, and I also, by the time people were coming to me with this question, I was simply told what their discharge limit is there. I'm certain that there's some reason for that, but I'm with you that natural waters, rivers, plenty of other lakes around here that I've sampled, have a pH that comes close to 10 regularly.
The lakes that are that are almost like you would say over, right?
Yeah.
So, and then the other question is, have you done any day-night sampling of pH?
I did not have a chance to head out there for day-night sampling, and it is something that would be up to the Stillwater group to go ahead and sample. It's an interesting question there as to how quickly that pH shifts and whether or not there is a diurnal cycle going on with the algal growth there. I expect that there is, yeah.
Other questions? Thanks, Seth.